Hi, everyone. Good afternoon. Welcome to GS 182, which is the Introductory Biology. My name is Vincent Wong, and I will be teaching you the course. I've been teaching at Centennial for 12 years now, I think. And for the most part, during the 12 years, I have been teaching this course as well as the pre-anatomy physiology, which is what you will be taking in the second semester. So maybe I'll see some of you again next semester. Okay, so before we begin, I would like to just go through some basic information about the course, where to get information, where to download lecture notes, for example, and I'll tell you about the format of the assessment, when we're going to have quizzes, and so on and so forth. If you have questions at any time, please feel free to unmute and just ask the question directly. Or if you prefer to type the question in the chat box, that is okay as well. So sometimes I look like I'm not looking at the computer like this. That's because I'm looking at the other screen. That's where the chat box is. So I'll be monitoring your questions if you have any. So you receive an email from me, last week, and then a couple of reminders throughout the week. And pretty much in that email, I told you a little bit about what to expect in terms of the delivery. On your timetable, you have the Tuesday and the Friday slots, both 12:30 to 2:20. And so we will be doing asynchronous learning on the Tuesday slots, where you'll be, watching the lecture recording on your own. And then for the Friday, this is the time where we will come together and we will have a face-to-face lesson. Okay, so the pre-recordings, that's like the entire lecture. Okay, it's more like a full movie, if you would. And what we are doing in the tutorial, it's kind of like a condensed version of that, kind of like a recap or like a synopsis of the entire lecture. So ideally, you would watch that before coming to class. But for whatever reason, you didn't have time to do it. Don't feel like you cannot come to the tutorial. Okay, come regardless if you have watched it or not. Ideally, you have watched it, but if you haven't, please still come to the tutorial, because we cover a lot in the tutorial. We take up the study guide. We will do some practice questions, and then there will be some chance for you to ask questions and interact with me a little bit as well. Okay, so having said that, let's just do a quick poll and see if you have watched the recordings or not. It's an anonymous poll, so feel free to tell me the honest answer. If you have watched the recording before you came, please choose A. If you haven't had a chance to do that yet, please choose B. And if you don't know where to find the recordings, you can choose C. Okay, so that's pretty good. 70% of you have watched it, and then some of you are not sure where to find it, or you haven't had a chance to do it. So let me quickly go through how to get to it. First of all, the easiest way to find things is just to look at the announcement I post. Okay, all these blue things, they are called hyperlinks, and it will take you directly to what I'm referring to. So when I say download scientific method lecture, you can just click on that. Okay, and then watch the recording. You can click on that, and it will take you directly to that page. However, if you cannot find the specific announcement that have the hyperlinks in it, you can always click on content on the top left here. And you have 3 folders here: the lecture folders, the virtual labs, and the useful information folder. And you want to go into the lecture one. And the first lecture is the scientific method. So under the scientific method folder, you have 3 other folders: lecture recordings, study resources, and supplementary readings. So if you click on it, you will see this is the PowerPoint slides. Again, if you have a printer, you probably want to print that out so you can take notes while you are watching the recordings. I try to keep the recordings to under 30, 45 minutes so that you would have a little bit of a break. Watching it all at one go might be a little bit onerous or intense. So you might want to watch them in small chunks and have time to digest the material. And then you come to the tutorial, like I said. And then under the study resources, you want to get the study guide. So the study guide is what we will be taking up during the tutorial. You are welcome to try them out on your own first, and then bring it to class, and then we can take it up together, ask questions, because the study guide is what will help you prepare for the quiz and for the test. Okay, so if you have a printer, perfect, print it out. If you don't, you can just leave it on your computer and type the answers in as we are doing it. Sometimes the lecture recordings require you to have the study guide. Okay? There are some parts of the recording where I will be referring to the study guides and we'll be doing questions as we go. So make sure you always have a copy of that. There is a video resource here. So if you click on that, it will take you to, this is called Padlet. I don't know if you've seen a Padlet before, Padlet, that's what they're called. So it's a collection of videos that I found throughout the internet that relates to the topic that we are talking about. So sometimes you might find it useful to listen to explanation from a different perspective, right? And sometimes you have I would just have some interesting videos that I found that is relevant to a topic, and then you can watch them. These are not mandatory. You will not be tested on them. Oh, by the way, this rarely happens, but it happened this morning. Couple minutes into the lecture, my computer froze, and I wasn't able to come back until like 10 minutes later. Okay, so if for whatever reason I get disconnected, please just stay on the Zoom link, and I'll come back as soon as possible. I will come back, okay? In the unlikely event that I cannot come back for whatever reason, like I get kidnapped or something, I'll try to post an announcement on eCentennial, and I'll send you an email letting you know that I won't be able to come back. But hopefully that will not happen. If you get disconnected, feel free to come back at any time. And if you cannot come back and you miss the entire tutorial, that's okay as well, because there will be a recording that I will post afterwards, and you will be able to watch it. Okay, so that's the video resources. If I go back here, you can see there are some crosswords you can do, and there is a self-quiz for each of the topic. The self-quiz you can do as many times as you like. They are not for grades. It's just a formative assessment for you to see how much you know and how much you need to study for. So feel free to take advantage of these things. There is also a supplementary study, and if you supplementary reading rather, if you click on that, it will take you to the online textbook. Okay, so one of the advantages of switching to online learning is I've decided to take away the textbook entirely. So you don't have to purchase a textbook anymore. You can just use these online free textbooks. And that will be the same for second semester when you take anatomy physiology. It's going to be the online textbook. Okay, and some people would like to do extra reading to help them understand more about what we're learning. But as I said before, any additional information that you've learned in the readings that we did not cover in class, you won't be responsible for them on the test. The only thing I will test you and quiz you on would be the lecture recording, the tutorial things that I go through with you, as well as the study guide. Do we have any question so far before I proceed? Okay, great. Again, feel free to jump in anytime. Let's see what else I have to tell you. All right. Go back to the home page here. You can always click on the name of the course to take you back to the original page. You want to download a copy of the weekly schedule. Okay, so you can just click directly on this button. I have a desktop copy that I'm just going to open. It's bigger, easier for you to see. So on Tuesday, you can see you will be doing the asynchronous lecture, watching the recordings, doing the study guide. You don't necessarily have to do it on that day, obviously. You can do it anytime. The advantage of having a class on the Friday afternoon is you're gonna have the entire week, right, to do those to watch those recordings before coming. So hopefully, for those of you who didn't have a chance to do that this week, you will be able to do it next week. So for next week, you'll be watching the ones about macromolecules. Okay, so for the most part, we will have our synchronous session, which is what we're doing right now. This is called synchronous, on the Friday, with the exception of whenever there is a test. Okay, so the first test is going to be week 7. And on that week, we will be doing the tutorial, the synchronous session on a Tuesday. Okay, so you can mark that down on your calendar. And if you don't mark it down, that's okay. I will be sending you reminders throughout the semester about these, these things. So check your email. Obviously, I communicate primarily through email, and also check eCentennial because I'll be posting a lot of stuff on there as well. For those of you who found a phone number somewhere, there might be like a phone number on some directory in the college to me. Try not to call me, okay? The best way to reach me is email. I reply email very quickly, usually within an hour. If it's during like, if I'm not sleeping, I'll reply you within an hour, okay? If you email me like 11:30 p.m. at night, then you might have to wait until the next morning, okay? But generally speaking, I reply your email very quickly. If you do not hear a reply from me for more than a day, that means I did not get your email, okay? So this is my email address. You can find it on the course homepage. There is another one that says vwong41. That one also works. But it's not at centennialcollege.ca. That one is at my.centennialcollege.ca. Both of them works, but there are other Vincent Wong in the college apparently, and sometimes for some reason people send it to the wrong Vincent. Okay, and so if I don't reply you, that means I did not get your email. This is very important because students sometimes tell me, I emailed you and told you I cannot access the quiz or like I cannot write the quiz, but I never got their email, okay? So as a general rule, if you don't hear me back from me for more than a day, please send again, okay? So what's gonna be the evaluations? Well, for the evaluations, the course is separated into 2 parts, two units, like, well, not unit, more like 2 parts. Okay, first half and second half. Okay, so for the first half, you are going to have quiz number 1 and quiz number 2. Each of them will be 10%. And then you will have a lab. It's a virtual lab. You don't have to come to the school to do it. This is a beetroot lab. That's going to be 10%. And you will have a test, which is worth 20%. So quiz number 1 is going to be lecture 1 to lecture 2. Quiz number 2 is lecture 3, lecture 4. And then beetroot lab is beetroot lab. And then test 1 is going to be lecture 1 to lecture 5 plus the lab. Okay. After you finish the first half of the course, you can forget all about it because the second half we start fresh. Okay. Quiz number 3, it's going to be again 10%. Lecture 6 and lecture 7. Quiz number 4, same thing. So you kind of see the pattern now, right? And then you're gonna have a microscopy lab, which you don't have to write this down. This is like all in the like in the calendar and stuff like that. So this would be what is it? Just the lab. And then test 2 is going to be 20% and will be lecture 6 to lecture 10 plus the lab. Okay, so the first half is kind of independent from the second half. That way you don't have to, like, there's no final exam, right? So you don't have to study for too much material. Hopefully this will help you do a little bit better. Any question about what I've said so far? Anyone? No? Okay, you guys know you can use the there is like a reaction button, right? Like in the Zoom the ribbon, I think it's what it's called, right? So like you can do like a thumbs up, right? Yeah, exactly. Some of you are doing it, right? So you can see if you click on the participant tab, then you can see that there is some reaction that you can do, right? Okay, like let me show you what I mean by that. Because sometimes I would just ask you, you see, so like some people like put like a thumbs up right here, right? Okay, so sometimes if I ask you just quick questions like yes or no question, then you can give me like a thumbs up, thumbs down. Then that way we can, expedite things a little bit. Next, you must be wondering what is going to be on the quiz and what is going to be on the test. So on the quiz or test, there are 3 types of questions I can ask you. Okay, so there is your standard multiple choice questions. These are 1 mark each, and it's your standard type of questions. You have A, B, C, D, E, or A, B, C, D, E, F sometimes, and you choose the correct answer. Right? So those would make up, I say, 70% of all the questions. Right? So straightforward stuff. Then you have what's called multi-select. Okay, these are 2 marks each, and they have more than 1 correct answer. And in order for you to get 4 marks, you have to choose all the correct answers. So that requires a little bit of, judgment call and thinking involved in these kind of questions. And then the hardest type of question would be the application question. Okay, it's still in the form of a multiple choice question, but these will require you to apply what we've learned in class in a scenario that it is new, okay, that you have not necessarily seen in lecture, but you would have the knowledge to solve the question. Okay, so the multi-select and the application questions are going to make up like 30% of the test. And before each quiz or test, I will post on eCentennial to tell you exactly what it's out of, how many of each type of questions you need to expect, and the time limit. Okay, so time limit, it's about, I would say 40 to 45 minutes for a quiz, and for a test, probably like around 70, 75 minutes. If you require accommodation, if you have a IEP from high school, or if you require accommodation for extra time and you're registered with CALS, C-A-L-S, that's the Center for Student who requires accommodation, please have them forward the document to me, and you can contact me individually as well through email, and I will accommodate you accordingly. When will the quiz and test be happening? It will happen on the day that is specified on the calendar. So your first quiz is going to happen in week number 3 on the 21st. And typically the quiz or the test will be available for from 10 a.m. to 10 p.m. And once so you can log in, you can access the quiz anytime during the period. But once you log in, you would have to finish the quiz during the login section. So like, let's say the quiz is like 40 minutes, right? So, and then you log in at 11 o'clock, then you would have to finish by 11:40, okay? You cannot log in and then close it and then come back at like, 2 p.m. and try to do it again. The clock, once it starts, like the timer, once it starts, it does not stop, even if you close the browser and stuff like that. So make sure when you do the quiz or the test, you are in a place where you're not going to be interrupted easily, and you would have, the entire duration to finish it. Now, sometimes problems occur, like your computer crash, your internet is gone, and you cannot log back in. That's okay. Right? It's understandable. Okay, you just have to send me an email, and then I will let you rewrite it or something like that. Okay, but if every time there is a quiz, you send me an email and say your internet is got disconnected, which actually happened like 2 semesters ago. So every time there's a quiz, this student would email me and say, the internet get disconnected. Then I get a little bit suspicious, right? So it's an honest system, right? Make sure you don't exploit it. And then there is the lab. Okay, so the lab you actually have access to the labs right now. Okay, so I'll show you where to get that. The lab, you would go to content, Okay. And it's okay if you don't follow everything I said right now, right? Like I told you, there will be a recording that you can watch later. So content, and then you want to, I'm just going to click on table of contents, and then you will have the virtual labs right here. So there are two labs, which is the beetroot lab and the microscopy lab. And you can click on it and see what it looks like. So basically, you would have to download this lab onto your computer, and then obviously you read all these things, watch whatever I tell you to watch, and then you will have to answer the questions. So what is the purpose of the experiment? So you would download this and you would type the answer, and after you're done, you will save it and you will submit it. Okay, so if you feel like it, right, at the beginning of the semester, the workload is not too much yet. So if you want to get a head start, you can go ahead and at least finish some of the lab, but you won't be able to hand it in until later. Okay, so to hand in the labs, you want to go to assessment, assignment, and then you will see there are two folders. One folder for beetroot experiment, one folder for microscopy. They are not open yet. They will be open on the date that is shown here. Okay, so you can finish the lab first, save it, and then when it's time to submit it, you can submit it. Okay, and if you submit it and then you decided to make a change, you can submit another one, but then the original one will get overwritten. Okay, so you would have from October 4th all the way to the 8th, that week you will submit the lab, and then afterwards I will close the Dropbox, and then I will mark your assignment. Does that make sense? Does anyone have any question? Okay. What else do I have to tell you? Let me see. I think that's it. Oh, this yellow button, right? You can click on that, and it will take you to this page. This is going to be the link that you're using right now. And there's nothing right now, but then after class, I'm gonna post today's recording. And then next week, I will add the next week's recording, and so on and so forth. So if you ever need to go back to a particular recording, this is the place to be. Okay, again, you can just click on the yellow button to get there. Christine asks, oh, sorry, Christian asks, will all of these labs and such be online? Yeah, everything is done online. Okay, there is absolutely no need to go to school, to go to campus for this course. And everything you need for the entire course is already up there. Okay, so if you've done biology in high school and this is just a refresher for you, you might want to go ahead and watch lecture 2's recording, lecture 3, and so on and so forth. Okay, last semester I had someone who like finished watching all the recordings in week 4, okay, because she has done biology before. And if you haven't done biology before, or like you have been out of school for some time and you're taking this bio course for the first time, that's okay as well. Okay, my idea is I assume nobody knows any biology, so I'm going to start at a basic introductory level. So that will be the common denominator for all of us. Okay. Anybody have anything else before we get started? So I have a question for you. If you have a copy of the study guide, which is what's being shown on the screen right now, please choose A. If you do not have it, please choose B. If you don't know where to find it, please choose C. Okay, very good. Again, 70% of you have it. So for those of you who do not have it, this is how you get it. And if you have access to a printer, please print it like maybe now. But if it's not easily accessible, the printer, then get some paper and pencil, and then you can copy what I have. Okay. So the quickest way for you to get it now is just to go to the homepage, And then under the announcement that I post, you want to click on this one. Okay. Study guide. Okay. Study guide. Normally, you can access it through the content, the lecture, and then the study resources. Okay. So normally, just one more time very quickly, content, and then you want to go to lecture, choose the lecture that you're doing, study resources, and right here, study guide. Okay. So if you can't print it, just have an electronic copy and type it in as you go. Okay. So I won't be posting the filled in copy of the study guide. If you miss anything, again, you can just watch the recordings and it will come up. Okay. All right, let's get started. In the first lesson, we talked about the scientific methods, but before we did the scientific methods, we also talked a little bit about, living things and then, how science is being conducted. So let's start with the characteristics of living things. Okay, so the first characteristics that I remember, this is not in any particular order. Okay, it might not be in the same order as the lecture notes, but doesn't really matter. So first characteristic is that they exhibit order. Okay, so e.g., for example, there are structures, there are patterns, symmetry. Okay, there is some kind of orderly architect to the living organism. If you think about like animals have left-right symmetry. Okay, if you think about flowers, there are patterns. Living organisms, they have various structures that are specialized in doing a specific function. Okay, so all living things will have some kind of order. Second one is the ability to maintain. Let me know if I'm going too fast, okay? Ability to maintain homeostasis. Can someone explain to me what is the meaning of homeostasis? You can unmute or you can type it in the chat box. Just gonna give you guys a moment. What is homeostasis? So people say balance, internal balance. Yeah, so most people got it. The homeostasis is the ability to maintain an internal balance. So the most like the simplest example to demonstrate this is humans. We are warm-blooded. That means we maintain an internal body temperature of 37 degrees Celsius, and that is homeostasis. In order to maintain homeostasis, there are different ways or mechanisms that the body could use. So we can either use physiological, mechanisms, or it could be behavioral. Okay. So I don't think there is an I, sorry. So physiological are things that your body does automatically. Let's say it's very cold outside. A physiological response to that would be you shiver. Okay? Shivering is going to bring up your body temperature, and then you're gonna, maintain that 37 degrees Celsius. An example of a behavioral response to cold temperature is you put on a jacket. Okay, so one is your body does that automatically. The behavioral aspect, it's something that you've learned from experience. Next one. Next one is growth and development. Okay, so we were very tiny when we were babies, and then now we grow and we develop. Okay, other organisms have very distinct growth phase, like a butterfly, right? Start with the egg, the caterpillar, the cocoon, and then the butterfly. Very distinct stage. Another characteristic is requires energy. Okay, so animals, we consume food to obtain energy. Things like flowers, plants, they would take the energy directly from the sun and do photosynthesis. We are also, the living organisms are also able to respond to stimulus, okay, or stimuli, which is the plural. So a stimulus or a stimuli is basically a change in the environment. Now, sometimes the change in environment will prompt a physiological or behavioral response such that the body is able to maintain homeostasis. So in our example, when it's cold outside, that's the stimulus, and then you shiver, you put on jacket, that's a response, and it will help you maintain homeostasis. What's another one? Anybody? Anybody remember another characteristics? And feel free to unmute. Yeah, so some people say reproduction. Okay. Living organisms is able to reproduce and create more of their own kind, right? So humans make more baby humans, pandas make more baby pandas. You know, sunflowers will make more sunflowers. Okay, so they reproduce and make their own kind. And finally, they are able to adapt and evolve. Okay, it's important to remember that evolution, right, is talking about the population. Population evolve, not individual. Okay, so let me just give you a very, very brief crash course explanation of evolution. Let's say there is an organism, okay, and these organisms have two versions of them, okay? So some of them have good long fur, others don't, okay? And these fur is a genetic trait, something that can be passed on to the next generation, okay? And let's say all of a sudden, one day, the temperature becomes very, very cold, okay? Minus 70 degrees. Okay, let's say there is an ice age coming or something. So what happens is the organisms that does not have the thick coat, the fur, they're gonna die from the cold, okay? Some might survive, maybe they find a cave or something, but a majority of them would die from the cold weather. And then these furry ones will be the ones that are left behind. And then these furry ones are going to be able to produce more babies, and the babies that they produce, right? The babies that they produce will also have the fur, because the fur is the genetic trait that can be passed on, right? So over several generations, you will have fewer and fewer of these naked ones, so to speak, and then you will have more and more of these furry ones. So you see the population has changed over time so that they are more adapted to the minus 70. Now, individuals do not evolve, meaning this little fellow right here, if it's very cold, it's not possible for this particular one to all of a sudden grow fur and be adapted to the environment. Okay, it's the whole population that's going to change. Okay, if the temperature all of a sudden becomes very hot, like plus 70, then the furry ones will not be doing so well. Maybe they overheat and they die. And now these ones would prevail, and over time, the population would change again. So evolution is this ability for the population to become more adapted to a particular environment. There's a question. Is respiration a characteristic of living things? Respiration is a loose term. Like, what do you mean by respiration? As in like breathing, or like as in like cellular respiration? Okay, maybe I'll wait for you to type the answer later, but not everything does cellular respiration. There are some organisms that doesn't necessarily do that. If you mean respiration as in breathing, then some organisms don't necessarily breathe. So generally, that's not a characteristic of all living things, but a requisition of energy, that would be it. All right, moving on. We have two words that are very confusing for a lot of people: induction reasoning and deduction. Okay, so let's start with the meaning of each, and then we are going to come up with some examples to illustrate it. Okay, so let's use red here. Induction. Okay, so basically, induction means to derive, to come up with, derive a general statement based on many specific examples or observation. So this goes from specific to general. So you take many small observations, okay, like one observation, two observations, three observations, four observations, and many more. You take all of these, and then you come up with general statement, generalization. So an example, simple example would be something like this. Fish is made up of cells. It's made up of cells. Birds are made up of cells. Trees are made up of cells. Okay. Bacteria made up of cells. So you make a bunch of these observations, and then you generalize, and you're like, oh, all living things are made up of cells. Okay, so we take many small observations that are related to each other, and then you come up with a broad statement. That's called induction or inductive reasoning. Okay. Deduction is the other way around. In this case, the reasoning, probably should use a different color. The reasoning flows from general to specific cases. If you cannot read my writing, just let me know. I know I don't have the best handwriting. So here you will take the general statement, and then you will apply it to different situations. Okay, situation 1, situation 2, 3, 4. Okay. Example. So a related example would be, you start with all living things. All living things consist of cells. And then you would apply it to one situation. You'd be like, this this furry thing that I was talking about, right? This is a living thing. Therefore, it is made up of cells. Okay, and then let's say you find another living thing. You're like, oh, this is another living thing. It must also be made up of cells. Okay. A more relevant example, I guess, is general statement. Electronics run on electricity, runs on batteries. Okay, and then you find a calculator. Calculator is an electronic device. It must run on batteries. Okay. General to specific. Any question about induction and deduction? Okay, let's see how you guys do in this question. This is an example of something you might see on a quiz. Please give it a moment and try it out. Oh, the question is on the screen. Okay, the question is on the screen. I am sharing my screen, right? You guys can see the multiple choice question here, right?
Yeah, we're good.
Okay, thanks. Oh, that's okay. If you accidentally choose the wrong answer, don't worry about it. You can just think about what the actual answer is, and then I'll take it in a bit. Okay, maybe just another 15 seconds or so. Okay. So here are the results. I just shared the results with you. And looks like it's kind of all over the place, but there are two kind of popular choice, A and C. So let's talk about this one a little bit more. Okay. Where is this? Okay, here. So which of the following is an example of deductive reasoning? So deductive reasoning goes from general statement and you're going to apply it to specific cases. So let's take a look at the first one. All virus requires a host to replicate. Okay, all virus requires a host to replicate. This is a general statement. General statement. COVID-19 is a virus. It must require a host to replicate. This one is specific. Okay, so using a general statement, we apply it to a specific case. That is deductive reasoning. So that is the correct answer. Let's take a look at the other ones and see why they are not. B, COVID-19, Ebola, influenza are viruses that requires a host to replicate. Therefore, all virus requires a host to replicate. This one goes from specific to general. So this is actually deductive. Sorry, this is actually inductive reasoning. Induction. Okay, so this is like saying the fish, the bird, the flowers, they're all made up of cells, right? And therefore, all living things are made up of cells. Right? So this is actually not the answer. Now, C is a little bit tricky. At first glance, it sounds exactly the same as A. Okay, so you start with a general statement: all viruses requires a host to replicate, and then COVID-19 requires a host to replicate. Therefore, COVID-19 is a virus. The problem with this is, even though it seems like we're going from general to specific, but then the logic is flawed in this case. Let me do another example that sounds exactly like this, but using something that's a little bit easier to understand. So instead of saying all virus requires a host to replicate, I'll say all birds. If birds are like the virus in this case, all birds have wings. Okay. And then the next statement said COVID-19 requires a host to replicate, right? So it copies like the second part of the sentence, right? So instead of COVID-19, I'll say bees have wings. Okay. So according to this logic, therefore what? What am I going to say next? Finish the sentence for me, someone.
Bees are birds.
Therefore, bees are birds. And we know this is not true, right? This logic is flawed, right? And that's exactly what the logic is here. Okay. So for that reason, this is not true. Okay. It has to be the other way around in A. And D is not correct because we are just taking one specific example and then apply it to another specific example. There is no general statement going to specific cases. This is just one specific case to another specific case. So that is not, while there's nothing wrong with this, it is not an example of deductive reasoning. Okay, so the correct answer for this is A. Any question? So you might see something like this on a quiz, okay? This would be like maybe like an application question. We all clear on this? Perfect. Okay, list the steps in the scientific process. So it's just the steps that are in the PowerPoint. I can write it down for you here quickly. We have observations. Okay, so you're going to make many observations, you know, either through measurements or through using your senses, and then you're going to ask questions. And then to try to answer your own question, you're going to form a hypothesis. Okay, so a hypothesis is a tentative answer, right? Some people call it an educated guess. I like tentative answers more. It doesn't have to be a guess. Now, for a hypothesis to be valid, it needs to be testable and falsifiable. Okay, it has to be potentially wrong, falsifiable. Okay, potentially wrong. After you make a hypothesis, you are going to make a prediction of the results. Predict results. You're going to do an experiment to check those predictions. And then you're going to make a conclusion to see whether your hypothesis was accepted or if it was rejected. Okay? If you reject the hypothesis, you're going to have to make a new hypothesis, and you will have to conduct a second experiment, so on and so forth. Okay, so that is that. Now, let's give an example of a non-testable hypothesis. Okay, so something that cannot be tested would be like chocolate tastes better than candy. Okay, that is not testable. That is a subjective thing. depends on the person. It's an opinion. It's a personal choice. There's no right or wrong. Cannot be tested. So this is subjective. Okay. It is an opinion. You cannot, scientifically test an opinion. Does anybody have an example of a non-falsifiable hypothesis? Something that cannot be wrong. There is no God. Okay, yeah, that's one example. Good. You cannot prove that true or false in one way. 2 + 2 = 4. That's an interesting one. That's more like a definition, right? We don't really, we cannot, like, it's a accepted definition. 2 + 2 = 4, Daniel says. So if something that is an accepted definition, we wouldn't, call it a hypothesis. But interesting example. I have one. Okay, if you work hard enough. Sounds like something my parents would say to me when I was younger. Where if you work hard enough, then you will get A+ in biology. Okay. So this one is not falsifiable. It's not falsifiable. Because if I don't get A+, if I only got B or C, then the counter argument is that, well, you didn't work hard enough. Okay, and then if I work harder and I still only get a B, and then the person can still say, well, you didn't work hard enough, right? Yeah, that's my parents basically growing up. Story of my life. Okay, so essentially, the statement can never be shown to be false. Okay, so Ronaldo is better than Messi. Okay, yeah, again, that's an opinion, right? That's not testable. Okay, potentially is not falsifiable as well. There are a couple other ones that here. All living thing needs water to survive. That's an interesting one. If you phrase it that way, it's hard to prove it false, right? But it sounds like it cannot be proved false. But if one day I discover a living thing and it's able to survive without water, then I would show that hypothesis to be untrue, and therefore it is falsifiable. Okay, so for example, let me put it another way. If I say all boys, I don't know why I came up with this example. All boys don't eat. I don't know. What do people don't eat? Pizza or something. Okay. Let's say I make a statement like this. The only way for me to show that this is not true is I find a boy and then I give him some pizza and then he eats it. And then this statement would be shown to be false, right? So that's what it means by falsifiable. Okay, falsifiable means if you can find just one example to counteract the statement to show that it's untrue, right? Then that's good enough. Okay, and that's the idea of having a falsifiable hypothesis. Okay, if you have a hypothesis that can never be proven to be untrue, then you cannot conduct an experiment. Okay, so that's what it means. Okay, hopefully that makes sense. If not, anybody have any follow-up questions? You guys still with me? Has been an hour of me talking. Are you guys still there?
Yes,
Friday
sir.
afternoon class. Thank you for coming, actually. I know it's not the best time, but we're all here. And actually, attendance is fantastic. Almost all of you came, which is great. Okay, moving on. Give 2 examples of primary sources and 2 examples of secondary resources. So generally speaking, primary resources are more reliable because there are many layers of safeguarding, and usually the information are vetted before they're released, so they're more reliable. Secondary resources, some of them are pretty reliable, but there are also many misinformation out there and biased information. So example of primary sources are peer review journals. Okay, so if you are a scientist and you did some research and you want to publish your results, you would submit your work to a particular journal. There are many scientific journals out there. You might have heard of some of it. There are some called Nature. There is the British Medical Journal. All these different types of journals. And then other experts in the field will look at your work and give you some criticism. Okay, it will suggest improvements, or they would tell you that this is not working, and then you will have to make the adjustment. And if it meets the standards of the committee, then you can publish the results. Okay, so something like that has an extra layer to ensure the standards, and usually it's quite reliable. Okay, however, that's not always the case. Okay, one example that comes to mind is there is a person named Dr. Andrew Wakefield. Does anybody know who that is? Anybody heard of that name, Andrew Wakefield? No. Okay. So he published a paper in Lancet. Okay, Lancet is a very well-known journal, one of the top ones. Okay, and in that report, okay, he said that there is a link between the MMR vaccine, the measles, mumps, rubella vaccine, and autism in children. Okay, and that actually is the reason why we have the anti-vaccine movements. Okay, that started the whole thing about vaccines causing autism and its negative effects and all that stuff. And then it turns out later on that the results he published was not true. Okay, he actually made up the results. Okay, there was some conflict of interest about who was funding the research, and then the journal, the Lancet, actually has to pull the paper off and say, this has been retracted. Okay, this is not true, but the damage has already been done. Okay, many people still cite that article saying, vaccines are not good. And as you can appreciate, in the middle of a pandemic, these anti-vaccines, situations is actually doing a lot of harm, right? So even though peer-reviewed journals are, for the most part, reliable, sometimes these things do happen. Another example is a journalist reporting live from a scene. Okay, so you remember there was a tornado in Barrie, right, a couple of weeks ago, maybe last month, right? And you see journalists reporting live from the scene. That is primary source. Okay, they are there. They are doing it directly from where it is happening. Secondary sources would be things like a blog. Okay, blogs are, anybody can post anything. I can write anything I like on a blog and put it on the internet. There is no vetting process. Anybody can read it. And so, it could contain misinformation. It could contain wrong information. It could contain biased information. Okay, it could be a textbook. Now, textbook, although it's a secondary resources, but generally speaking, textbook are relatively reliable, right? Okay, you read something from the bio textbook, you're not really gonna doubt that. Oh, here it says all living things are made out of cells. You're not gonna be like, that's biased. Okay, like who's gonna back that up, right? Typically, we trust textbooks to a certain degree, but it's considered as a secondary sources because the knowledge that's presented in the textbook typically arise from things like peer-reviewed journals. Okay. Now, this next part I actually already did in the lecture recording, so I'm not going to repeat it, but I will explain the difference between independent and dependent variable one more time. Okay. So for the independent variable, okay, independent variable, I'll do it right here. Independent. Okay, so this one is the variable that is being changed or manipulated purposely. Manipulated by the researcher on purpose. Okay, so when you do an experiment, you want to change the one variable, okay, and then you are going to see if this will affect the dependent variable. Okay, so independent variable is the one that you change, you are in charge of. Okay, that's how I can remember it, right? Independent, you are in charge of it. Whereas the dependent variable, right, dependent, okay, so this one is measured or observed to see if it is affected by, sorry, this is very messy, the independent variable. Okay, so let's say I change the temperature of the room. Okay, that's independent variable, and then I want to see if that's going to affect how my plants grow. Okay, so how they are growing, that would be the dependent variable. Okay, so by deliberately changing or manipulating the independent variable, you will then assess, measure, or observe to see if the dependent variable is affected. Sometimes it has no effects, sometimes it does. Okay. So here are a bunch of examples, and you know, it's in the recording, so I'm not going to go through them again. Okay, next we have some experiment stuff here with Rady and Pasteur. So let's take a look at the first one. Explain the spontaneous generation hypothesis. So spontaneous generation states that spontaneous generation states that living things come from non-living things. Okay, living things come from non-living things. Okay, so in the past, people are not sure where, you know, living things come from, and they say, oh, maybe the living things will arise from non-living things. So specifically, there was an observation that, you know, you and I are familiar with probably. If you leave a piece of meat outside, come back to a couple of days, there might be maggots and flies crawling all over the meat. Okay, so that's the observation that Rady and many people make. Okay, so what observation was Rady's hypothesis based on? It is that when a piece of meat is left outside, flies and maggots appear. Afterwards. Okay. Now, something so simple like this, you know, you might be laughing or thinking like, why do you even have to do an experiment on that? Like, obviously, if you put rotten food out, put food outside and let it rot, then flies and maggots are going to appear, right? It's just common knowledge. But remember, this is like, you know, centuries ago. So, you know, back then, it's not entirely apparent of what's going on. And another observation that was supporting spontaneous generation is that fish and frogs would sometimes rain from the sky, and people would be like, oh, the sky, non-living, it's creating these living things, the birds and not the birds, I mean the fish and the frogs. It's just raining down. Okay, now we know the reason for that. And actually it still does happen sometimes in nature. The reason is because there might be like a hurricane or like some kind of tornado that swept through the nearby bodies of water, and it scoops up all these fish and frogs along the way and send them flying inland. But back then people didn't know, and they thought that's how living things are created, okay, from non-living things. So to test the hypothesis, Rady did an experiment, okay, and I explained this experiment in the recordings, but here we go again. He divided the stuff into 3 groups, okay. So he has 3 jars. Group number 1, group number 2, and group number 3. And in each of the jar, he would put a piece of meat. Okay. Maybe some steak inside, some pork chop. I don't know what he used, but he put a piece of meat right there. And the first one, it is an open jar, okay, plus meat. Okay. Maybe we skipped the plus meat, like obviously there's a piece of meat there. The second group, he put like a mesh on top. Okay, so jar with netted lidded cover, netted cover, sorry. And then group 3 completely seal the jar. Okay, so sealed jar. Okay, so he has the 3 groups, and then he leave the jars outside and then wait for a couple of days. Okay, so in this experiment of his, can somebody tell me what the independent variable is? Anybody? What's the independent variable? Megan said the lid. Yeah, pretty much. Okay, so the type of covering, right? No cover, netted cover, sealed. The dependent variable. What about the dependent variable? What is it? Okay, so people say the meat is the dependent variable. Anyone else? Yeah, the presence of flies and maggots, right? Okay, so the meat is not a variable actually. It's just sitting there. The dependent variable, it's whether there are flies or not, right? So what were the results of Rady's experiment? So group number one, well, as you would expect, there are flies and maggots all over the place. Okay, so flies plus maggots. Number 2, jars with netted covers. You would see the flies up here, and then you would see the maggots crawling on the net. Okay, so flies and maggots on netting. Okay, and number 3, as you would expect, no flies or maggot. Okay, again, this is obvious, right, for us, but not so obvious long time ago. So what conclusion did Rady make? Meat does not produce flies or maggot. Okay. Only flies produce flies. So that's his conclusion. All right. So you might think, oh, case closed. Spontaneous generation has been proven to be untrue. However, not everybody accepted this. Okay. Some people say, while spontaneous generation might not be true for bigger organisms like the flies and the maggots, it is true for microorganisms. Okay, so that prompted Louis Pasteur to perform his experiment. Okay, so Louis Pasteur is also the same guy who invented pasteurization. Like the milk is pasteurized, right? If you drink honey, honey is pasteurized. There is also the raw ones, okay, but then most honeys are pasteurized, right? That means it has been killed off. All the bacteria has been killed off. Okay, so he also performed a set of experiment, and the reason he did it is that some people believe spontaneous generation might be true for microorganisms. Okay, even though it might not be true for flies and maggots, for bigger things, it might be true for microorganisms. So he then go on and do an experiment, okay? And in his hypothesis, he states that if cells arise from non-living things, then they will appear. They will appear in sterile broth. So broth is like, nutrient soup, right? Okay, like chicken broth, beef broth, or whatever. And sterile means it has been treated with heat to kill off everything. Okay. Moving on. So this is his experiment. Okay. He is going to have two groups. Okay. One with a flask that's open directly to the air. The other one is connected to a swan neck top. And so the group that does not have the modification, we call that the control group. Control group. And then the one with the swan neck top, this is the experimental group, or sometimes also called the treated group. Okay. So let's try that one more time. What is the independent variable in this experiment? The independent variable is the one that you're in control of. And in this case, it is the type of, the type of flask used, either an open top or a swan neck flask. Okay, and the dependent variable is to assess whether there will be microorganisms in the broth afterwards. So how did Pasteur ensure the initial conditions of all the flasks were identical? Well, he treated the flask and broth with heat. Okay, so as I mentioned earlier, it kills off everything. So if he looks at the broth, rather, he looks at the broth a couple of days later, and he finds that there are cells moving in them, then they know that it wasn't from before. These are new cells that are being formed. So what were the results? Well, as expected, no microorganism are found in the experimental group. Only in the control group. Okay, so the control group with the open flask, all the microorganisms in the air will fall right through the hole. It's gonna multiply in the nutrient broth and then turns it moldy or something. And then the other one, because because of the swan neck, right? So all the microorganisms in the air, they might land here, but they cannot climb up. So the broth will remain sterile. So the conclusion here is that he definitively proved that living things cannot arise from non-living things. Okay, so living things do not arise from living things. Okay. Any question before we move on? Okay, let's do another poll. Let's do another poll. Here is a question for you to try. Go ahead. Okay, a couple more minutes, seconds for people to put in their answers. Okay, I'm gonna stop there. Thank you for everyone who have participated. And if you didn't have a chance to try that yet, you can try it later as well. So 1 ml of the experimental drug is diluted with saline. Oh, sorry, that's not it. Where is the? Okay, hold on. Let me just copy the question here. Diluted in saline, and we want to know which one is a good control. And so first of all, we're trying to test the side effects on pregnant mice. So anything with male mice would not work, okay? Because they don't get pregnant. And non-pregnant mice would also be not suitable for our purpose, okay? So if the experimental group is the one receiving the drug, the control group should not be receiving the drug. So this one would also be incorrect, okay? And the proper control would be just pregnant mice injected with saline, okay? Because the Experimental drug is diluted in saline, right? So you're giving them the same kind of solution, but without a drug. Okay, so most of you got that right, which is good. Okay, let's go back to here. Okay, so we're going to do this question now. Okay, and I think I copied it right here. You are a lead scientist in a study to determine if taking multivitamin supplement reduces the risk of cold infection. Okay, so we want to write a hypothesis for this. Okay, we want to write a hypothesis for this. So let's do that. Okay, a hypothesis. There are only two logical hypotheses we can write. Okay, you either think multivitamin supplements will reduce the risk of cold infection, or it doesn't. Okay, and if you try to be creative, you can say, I mean, like, I don't mean it in a good way. Okay, you can also say multivitamin supplements will increase the risk of getting a cold infection, but you know that's probably unlikely. So the only two possibility is, like I said, taking multivitamin supplement will or will not, doesn't matter. I will say will reduce the risk of a cold infection. Okay, so that's my hypothesis. This is testable, okay, and it's falsifiable. Okay, it could potentially be wrong. So it's a valid hypothesis. B, what is the independent variable and the dependent variable? So the independent variable would be taking supplements or not. Okay, are they taking supplements or are they not? That's the independent. I can control that as an experimenter. Okay, I can decide who to give the vitamins to. Dependent variable, right? The thing that I'm going to be observing, measuring, assessing, that's going to be whether the people catch a cold or not. Catching a cold or not. Okay, so that's independent and dependent variable. C, what group should be involved in our experiment? Okay, so we should have a group of people, right? So people, right? Lots of people. Okay, so this one would be control group. Control group. So what does the control group get? Anybody? What does the control group get? Let me just make more people. There we go. What does the control group get for our experiment? What should we give them? We should give them the supplements. Okay, so I have various answers here. So control group are the people who does not receive any treatment. Okay, they do not receive the change. Okay, so in this case, nothing. Okay, so no vitamins. That's the control group. Okay, so the control group is the group that we will use to compare our results to. Okay, so nothing. Next, we have the experimental group. What should we give to the experimental group? That's the people that we are going to give vitamins to. Okay, the supplements, right? Okay. And the third group, okay, that's what Christian is suggesting in the chat, that is the placebo group. Can someone explain to me what a placebo is, please?
It's like you make them think that they're taking vitamins, but really they're taking something that doesn't do anything at all.
Exactly, like a sugar pill. Okay, so sugar pill. Okay, so you tell them, hey, have some vitamins, but it's actually just sugar. Okay, or like not sugar, but something that they cannot taste, right? Okay, like I don't know, like I cannot think of an example. Sorry, gotta my brain is stuck right there. But it's like something that doesn't have an effect, okay, on what you're trying to assess. Something that doesn't affect your chances of catching a cold. Okay, so the purpose of having a placebo is to account for, yeah, exactly, right? No therapeutic value. That is exactly what a placebo group is. So the purpose of a placebo is to account for bias as well as psychological effect. Okay, my daughter, right? I think I probably shared this in the video. Like she would fall down and then scrape her knees, not bleeding or anything, but then she would be crying. Oh, it hurts. I am not feeling well. And then I would give her a band-aid, and everything is going to be okay again. Right? Did the band-aid do anything like to physically help with the wound? Absolutely not. But psychologically, right, it makes her feel better. Right? It's a placebo effect. Okay, so we want to account for that. That's why we have to have a placebo group. Okay. Next one. What's the next one? Okay, so this next one is important. Okay, what factors must be kept constant between various groups? So I have 3 groups, and then I can only have one thing that is different between them. So no vitamins, sugar pill, and vitamins, and everything else we would try to keep as constant as possible. Now with the Rady experiment, right, it was easy to control. He could choose what meat to use, and as long as he's using the same pieces of meat for all 3, it is okay. He can choose the size of the meat, right? Has to be brought on the same day, for example. Size of the jar, all those things he could control. But it's a little bit more complicated when we are trying to deal with humans. Okay, we cannot have identical humans; they don't exist, right? We cannot just clone everybody. So we try to keep them as consistent as possible across all 3 groups. So these things that we're trying to keep consistent, we call them standardized variables. Okay, so I want you to help me out here in the chat box. Okay, and if you don't know how to use the chat box, like just mouse over the ribbon on your Zoom meeting. There is like a little speech bubble. You click on that, and then the chat box would pop open. Okay, I post a link on the chat box, and it will take you to this page. Okay, so I want you to list 3 standardized variables for this experiment that I'm talking about. Okay, what are the variables that we have to keep constant between the 3 groups so that the results would be as accurate as possible? Okay, if you do not see the link for whatever reason in your browser, you can type in this website, www.menti.com, and then you can key in the code, and then you will be able to participate. Okay, I do want you to participate though, so I'll keep the code here and I'll move that away. Okay, does everybody understand what I want you to do? So I'll give you a minute or 2 to try that. Okay, I have 2 people so far. All right, does everybody are everybody able to access the link? Maybe you guys just taking your time. If you can only think of like 2 or 1, that's okay as well. You can just submit whatever you have, okay? So maybe just a little longer for that. Okay, I see more answers coming in now, good. Okay, so keep going if you want. I will take a look at what people write. So, age is a good one. Okay, you want to keep people around the same age group in the control, the placebo, and the experimental. You don't want to have all the young people in one group and all the old people in the other group, because that's going to have different effects, right? So everybody should be around, say, 20 to 40, something like that, okay? Or at least have the same number of people from each age group in each of the group. So what that means is, in the control group, you want to have 100 participants from 20 to 30, 100 from 30 to 40, so on and so forth. And then you will have the exact same setup in the next group and the next group, okay? Diet is good, right? You want them to have similar diet, although that might be hard to control. You cannot, you cannot put everybody in a hotel and then force them to eat the same thing for the next, couple of weeks or something, or however long the study is. But that would be something to consider, okay? Because there might be some diet that would be, I don't know, rich in carbohydrates or something, and maybe that's going to have an effect on whether or not you're going to catch a cold, okay? Not so much the kind of vitamins, okay? Because only one group is receiving the vitamin, right? Which is the experimental group. When we talk about standardized variables, we are talking about things that we would want to standardize, that we want to make the same for each of the groups, okay? So yeah, sex and gender, they mean the same thing. So you don't want to just use the same gender in one group and then the opposite gender in the other. You want a good mix, maybe 50-50, okay? Not sure what some of these means. Stress level, exercise. Okay, so you want people to have similar physical, similar level of physical activities, right, during the duration of the studies, and you want them to. I think I saw something here earlier, like the environment or something. I seem to have lost it, but I thought I saw one. Yeah, like the exposure, right? Like you don't want to put healthcare worker in one group and then office workers in the other, because naturally the healthcare workers would be more vulnerable to catching a cold, right? So you want to have a good mix. Okay, so does everybody understand what standardized variable means? Right? It is the things that you try to keep the same for all the experimental group. Okay, it's particularly complicated for people because it's you cannot find identical people, but you try your best to keep everything standards. Another one is ethnicity. I don't think anybody mentioned that, but that would be it. Good, very good. Moving on. What results would support your hypothesis? Well, if the people who receive the supplement end up not catching as much cold, then it would support your hypothesis. Otherwise, you would refute it. However, there is a catch, and I'm going to come to the catch later on. But for now, let's just keep it simple. Okay. Any question before we move on? No? Okay. Let's see. What's next? A farmer wants to determine if feeding cows with high protein diet would increase milk yield. So the hypothesis is again quite straightforward. You either say feeding the cows with high protein diet will increase milk yield or it will not. Either one is correct, it's fine. Okay, so I'm not going to write it out. It's pretty straightforward. And how would you set up the experiment? Well, I would have some cows, okay, some cows. And this is group number 1. It will be the control. Okay, and then we would have the exact same group of cows on the other side. And this one would be the experimental group. Okay, so the control group would receive normal protein level in their diet, basically the standard stuff that you feed them. And the experimental group would have high protein diet. Okay, and I would have to standardize the variables between the two groups, right? So I would have to make sure that these are cows of the similar age, they are raised on the same pasture, they are going to be eating the same protein, okay? But the levels would be different, okay? So they would have the same protein source, but this one would be higher, like more protein, and this one would be less protein. It's just the amount that's being varied, but not the type of proteins, right? And we want them obviously to be the same breed of cows, free of disease and all that stuff. And then I would go ahead and measure the milk yield, okay? So the milk yield is the dependent variable, while the amount of proteins, that is going to be the independent variable, the one that I'm going to be able to control. Now I'm going to ask you this one. Should we include a placebo for this experiment? Is a placebo needed? Why or why not? If you think we need a placebo, please choose A. If you think we do not need a placebo, please choose B. Okay, most people say we do not need a placebo. Does anybody want to explain to the class why we don't need a placebo for this experiment? Anybody? Okay, so Michaela and Megan say cow doesn't know if it's getting more proteins or not because the cows don't know the experiment is being conducted. Exactly, right? The cows don't know anything. You feed them, they're like, good food, I'm gonna eat it. Okay, they're not gonna know whether they're part of the control group or experimental group. There's no bias, there's no psychological effect. Okay, but they're not gonna be like, I'm gonna hold out on the milk production because I know you're testing me for it, right? There's no such thing. Which is why we do not need a placebo. Placebo is more for like human, right? Only humans are complicated, right? Humans are the ones that are coming up with all these psychological effects, okay? So we do not need a placebo. Okay, moving on. Describe the main principles of experimental design. So basically what we have been talking about, so have proper groups, okay? Control and experimental, that would be the minimal two groups. And then if you're dealing with humans, you need a placebo, okay? You want to have a blind, you want to have a double blind whenever possible, double blind experiment whenever possible. So a blind experiment is one where the participant does not know if they are receiving a placebo or the actual drug, okay? So you don't tell them. A double blind is where even the researcher themselves do not know what they're giving, okay? So that's to account for like bias in the researcher, right? As a researcher, sometimes we are biased because we want the drug to work. We want the experiment to work. So when we assess the outcome, we might be a little bit biased. And so in a double-blind experiment, I don't know what I'm giving and they don't know what they're receiving, and it's considered as the gold standard. Okay, it's only after the data has been collected, then you can cross-match the data with like a decoding sheet almost to figure out who got what, and then you can evaluate it objectively. So Christian has a comment. A placebo can be used, talking about the cow thing. A placebo can be used because maybe cows can be tricked into yielding more milk, assuming they do get more milk from eating more. No, I don't think so, because the whole premise of the placebo is to minimize bias and psychological effects, right? So the premise here is that we assume the cows, they're not, I don't want to say they're not capable of thinking, but not in the degree that they would know that we are doing an experiment on them, right? So for that reason, we do not have to include a placebo to adjust for that bias. I hope that makes sense. And other things that you have to consider is a sample size. Sample size, right? You want to do the experiment on a big group. The bigger the group, the more accurate the data is, right? You might remember when people were doing the experiment on the vaccines, right? You hear numbers like, oh, Pfizer's vaccines are like 97% efficacy, and then AstraZeneca, I don't know, 78% efficacy, right? But then once you started using these vaccines in the real world, all of a sudden those numbers start to go down, right? It's no longer 97, it might go down to like 90% or 85, right? That's because when you do it in the real world, right, so many more people are involved in the process compared to the initial sample size, right? Initial studies, maybe they have hundreds of thousands of people, but compared to the real world, we talk about millions of people getting vaccinated, right? So the bigger the sample size, the more accurate the numbers are going to be, okay? So when you do an experiment, you want to involve as many people as possible. But like there are financial constraints, and then there are also physical constraints as to how many people you can involve. But generally speaking, as big of a sample size as possible. It's also important to know that sometimes we're not always doing experiments with people, right? Sometimes you're doing experiment on cells, right? So if it's cells, then you can do like millions and millions of cells, and that's possible to achieve. And you want it to be like a randomized sample, right? You don't want to handpick people. In some cases, you want it to be randomized, and that adjust for unintended bias results. We talk about the importance of having a placebo, and we talk about blind and double-blind experiment. So let's do two application questions here. The graph below shows the effect of zinc lozenges in reducing cold symptoms duration. Identify additional information that are needed to improve the credibility of the results. Okay, so suppose a salesman come knocking on the door, and then it's showing you this graph. We got this amazing lozenges, and it's going to help reduce the cold symptoms next time you catch a cold. And you can see from our graph, the people who take the zinc lozenges has a fewer number of days where they exhibit the symptoms, okay, compared to the placebo group. Right? So what kind of question are you going to ask the salesman to validate this graph? Like, what additional information would you like to see to make this graph more convincing? Or like, are you sold? Like, give me this stuff, I'm going to eat it right now. Right? What are you going to ask the person? Okay. Try to type some suggestions in the chat, or even better, unmute. Do you guys understand my question?
Can you repeat it?
Sure. So this graph shows that people who take zinc lozenges will have fewer number of days with cold symptoms. My question is, what additional information would make this graph more credible and more believable? Because there are critical informations that are missing here. So Michaela said to see the result of control group. Yeah, that's good. So control group. What else? Anything else? Okay, standardized variable. Yeah, age, which is part of the standardized variable, good, but how big is the sample? Exactly, sample size. Okay. And maybe one more, anybody? A couple more that I could think of. Anything else?
Maybe how many pills you take or whatever.
Yeah, could be the dose, right? Did the placebo people how often do you have to take these pills? Was it double blind? Yeah, exactly. Right? The nature of the experiment. Okay, and then a few more that I could think of would be who paid for this experiment? Right? Where was the experiment conducted? Is it conducted by the same pharmaceutical company that makes the lozenges, or like is it a third party? Right? All these things would matter for me to determine whether something like this is credible. Okay, so that is that. Okay, next one. This one also confused a lot of people. So let's talk about this. Basically, we have a new drug that will help reduce jet lag symptoms. And in a double-blind experiment, we see that these are the results. So control group, 70% reported that they have jet lag. Okay, so what do the control group receive? Nothing. Okay, receives nothing. We'll say no pills. Okay, placebo group, they have only 40% of people reporting they have jet lag. This is sugar pill. Okay. When I say sugar pill, I don't literally mean sugar, right? I mean like just something that is generic, right? Non-therapeutic. And then here we are going to have the sleep, the new drug. Okay. So based on these results, what do you think? Do you think it is effective or not effective? Choose A if you think the drug is going to help with jet lag. B if you think it is not. And if you're not sure, you can choose C. Okay, very good. Most of you got it right. If you have watched the lecture recording, you probably would have got it right because I explained this already. But let's explain it again quickly. Okay, if you look at the control and you see that 70% of people reported jet lag symptoms, that's expected. Okay, like if I give you nothing, you're gonna have jet lag. Okay, which totally makes sense. And then ignore the middle for a second and you look at the pills. You give them the new pills and you see that only 40% has jet lag symptoms, right? So you see a reduction compared to the control. So if you only have the control in the experimental group, you might think that the drug is working. But we have to look at the placebo as well. Okay, so the placebo is, again, you give them a sugar pill and you're like, take this pill, it will help with the jet lag and you will feel better. Okay, so people take it, okay, and you can see there is a reduction in jet lag symptoms, right? Only 40% of people reported it compared to 70% in the control, right? And that tells you that the drug is not really working, because you could have given these people just a random thing, something that has no effect on jet lag, and you tell them that it's going to help, right? And it would achieve the exact same effect. So that tells you that the drug is actually just pure psychological effect. It has no actual therapeutic value. Okay, so in this case, it is not effective. For this to be an effective a new drug, you would want to see that the placebo is performing at a similar level as the control. Okay, so that is that. And that takes us to the last part here, fill in the blanks. Let's just go through these things. So first one, ability to maintain a stable internal environment that is homeostasis. Next, we have part of discovery science where we make generalization from many small observations. So that's going from small to general statement, and that is induction. The opposite is deduction, where you go from a generalization to specific cases. A hypothesis is a tentative, testable explanation for an observation. And for it to be valid, it must be testable and potentially wrong, or falsifiable. Variable that you can measure or observe, that is the dependent variable. And the one that is changed or manipulated deliberately, that is the independent variable. A sham treatment is called a placebo. A study where subjects, that's the participants, do not know if they're receiving treatment or placebo. We call that the blind experiment, blind study. And if the person conducting the experiment also do not know what they're giving, then it's double blind. A correlation is a relationship between two variables. And in the recording, I talk about the difference between correlation and causality. Okay, just because there's a correlation between two things doesn't mean one causes the other. A primary source, it's a researcher's original report. And if it's coming from books, news report, and magazines, those are secondary resources, generally speaking. Okay, like you can have a book that's primary as well if the author is the one who did the experiment. Okay, so that's it for today. Thank you all for coming again, and thank you for your attention. I will post the recording after class. It will take some time to process, but it will be there. And then for next week, do lecture 2 recording, watch the lecture 2 recordings, and I'll see you next Friday at 12:30. Okay, have a good weekend. Bye for now.